Well, hello everyone. Good morning and welcome to Ocean Expert Exchange. Scientists in every Florida school and in Jerry Foundation are excited to have you join our live webinar events with ocean science and technology experts. In this monthly series, we dive into marine science, we explore what's happening in the field, we talk about interesting careers and more. And today we'll be bringing to you our speaker, William Sample from Florida International University. But before we do that, we want to tell you quickly a little bit about our programs. The Scientist in Every Florida School is a free program that's housed within the Thompson Earth Systems Institute at the University of Florida. The mission of CEFS is to engage K-12 students and teachers in all of the cutting edge research by providing science role models and experiences like today that we hope will inspire future stewards of our planet. Our partner, Jerry Foundation, is a nonprofit headquartered in West Palm Beach, Florida, and the foundation supports and promotes marine science research and education. And they do many of these initiatives with their 65-foot research vessel, the RV Jerry. In case you missed it, the information from today's preview slides, we will want to remind you that you can submit your questions for our scientist by typing them in the chat box, and we will get to those shortly. So at this time, I will stop sharing in just a moment to introduce you to our speaker, Will Sample. Will, I'm stopping so you can share, and please take the floor from here.
Awesome. Thanks, Stephanie. Let's
You're
go
welcome.
ahead and get mine shared right here. Perfect. All right. So let's go ahead and get started then, so I can hopefully have time for lots of awesome questions at the end. Today, I'd like to talk to you a little bit about what my research as a PhD candidate is at Florida International University, and hopefully talk about some cool things that we use that to study bull sharks in the Everglades for. So we'll start with who I am. So I'm Will. I am in the last year, hopefully, of my PhD at Florida International University. I grew up in Florida, so I'm originally from Jacksonville, and I've kind of worked my way down the coast. So I did my undergraduate at Florida Atlantic University. I double majored in marine biology and environmental science, and then decided to pursue a PhD afterwards down here in Miami at FIU. And I've been involved in all different kinds of research, from the current stuff I'm going to talk to you about today in the Everglades to some work in South Africa, working with great white sharks, which really made me fall in love with the subject, to pretty much any other project I can get my paws on here, to working with like permit and sharks in the Keys. So we always help out with lots of different projects around here, and I've got a really wide array of experience to draw from for my own research. So one of the big things I think is important for research is the underlying questions of why we're doing everything. And so for what I'm going to talk to you today about, I think the question we want to look at is why animals are going to move. And there's a lot of reasons they could move for. It could be to get food, it could be to socialize, it could be to avoid risk, or for sort of that herd mentality of gambit of the group, just kind of a bunch of different animals together. So thinking about why those animals might be moving, and sort of what they get out of that movement is one of the things that I always want to be kind of thinking about as where we're going through these different research questions and the different things we can do with them. The other thing that's important is kind of what's happening in the system. So I'm going to talk to you today about the Everglades. And the Everglades is an estuary. And if you don't know what that term means, it's basically a place where saltwater and freshwater are going to meet and they're going to mix. And historically in Florida, we had this fresh water that was flowing all the way from Lake Okeechobee, kind of down through the Florida Keys. And as Florida changed, that water flow got directed, went east to Tampa, or east to Fort Lauderdale, West Palm, west to Tampa. And nowadays, we're kind of restoring that natural flow. So we have this freshwater and saltwater that are kind of mixing again. And the ways in which that freshwater and saltwater are going to mix is going to be really important for anything that lives in that system, especially a bull shark, because bull sharks can tolerate freshwater and saltwater. So beyond being an estuary, the Everglades is also a bull shark nursery. So little baby bull sharks will stay in this area for the first 2 to 4 years of their life because it's nice and it's safe and it's protected. So just like a nursery might be for kind of us, if you have, a child or a younger sibling, it's the same concept. A nursery is kind of a safe place for that little shark to be. And the place that we actually work and the research takes place is all the way on that far southwest corner of Florida. It's labeled FCE LTER study area, which is just the Florida Coastal Everglades long-term ecological research. So it's this big project we've been doing for over 20 years now, monitoring these baby bull sharks in this area. So how do we monitor them? How do we study these sharks? Well, I like to think of it as almost reading the diary of these animals, kind of seeing what they're doing on a daily scale or a monthly scale or even years. And the way we use that, and the way we read that diary, rather, is with acoustic telemetry. So acoustics has to do with sound. If a room has good acoustics, I mean, it's got good sound, kind of good sound qualities. So it's coming out, we have sound, that's the acoustic part, and then telemetry, which is how we're following these animals. So there's two kind of broad types for this. There's passive and there's active. So right here on this slide now, we're looking at passive acoustic telemetry, which is that we put a sound producing device on the animal. So the animal's now sending out sound. In this case, this is the shark. And we have receivers that are kind of stationed out in the environment. They could be anywhere from, Biscayne Bay to the Everglades, what we're talking about, that are just kind of listening for that sound. And if a shark ever happens to swim by one of them, we'll hear the sound and know the animal was there. And this will just passively collect data for months or months and years. So that's the passive type. The other type is that we become that listening station. So it's the same concept. The animal gets a tag in it that sends out the sound, and instead of waiting for the animal to swim by our listening stations, we'll go out in the field and actually become that listening station, and stick what's called a hydrophone off the boat. So it's just like a microphone in the air. Hydrophone listens for sound in the water, and we'll listen for that sound from the shark, and we can even follow it kind of the whole time it's moving around. And using both of these together, we can take that active acoustic telemetry and that passive acoustic telemetry and look at how these animals might be moving over different times. So active is really good at looking at a single day or 2 or 3 days while you can follow the shark the whole time. And passive is really good for monitoring over months and months and even years. And so using these two different types of acoustic telemetry, we can sort of read the diary of the shark and see kind of, what it did on a certain day, where it was hanging out and where it was going. And the way that we actually do this on these animals is, like I mentioned first, the animal gets one of those tags that goes inside of the body cavity so it can produce the sound. So that's what's happening here on that video on the left. We put they're maybe like the size of a flash drive or like your pinky finger or so, goes inside the body cavity of the shark. We do one quick suture, sew them right back up, and they heal incredibly well from this. So in that bottom photo there, this is actually a shark that we caught 5 months later. And I think what's really clear from this is that while I will never be a plastic surgeon, these animals heal incredibly quickly, so much so that we can almost barely even see the scar after 5 months. And at 6 months, we've seen that the scars are almost completely faded, like undetectable. So that's how the animals get tagged and are able to send that sound out that we can listen for. And those tags will last about 6, 7, 8 years or so. It depends. From that, then we start listening for them. So that's what that thing in the middle is there. That's the hydrophone. So it listens for any time that shark that we just tagged swims within 500 meters of it. So if it ever gets nearby, we'll know that shark was there. And then we go out later, we download the data. It works just like a cell phone or a wire or a Bluetooth speaker, just connects via Bluetooth to the laptop. We download all the data, which makes me hopefully very happy because it means I have cool things to analyze. And then we can look at kind of where those animals might have been hanging out. Now, as we're doing this, these are the different locations that we're actually monitoring. So, like I mentioned earlier, we're studying in the southwest corner of Florida, down in what's called the Shark River. And each of those colored dots on the map right there is a listening station. So anytime a shark swims by one of those, we'll know if it ever got within 500 meters, and we'll know which specific shark it was, and then the time and the date it was there. And so we can take that and look at how these animals are moving. And we've seen kind of two interesting things that I think are going to inform some other questions. So, like I mentioned, bull sharks can go between fresh and salt water, especially when they're juveniles. And so there's different reasons they might want to go to different places. The fresher it is, there's probably less predators. So they're pretty safe up there. It's nice and safe and fresh, and there's not really going to be any other big sharks. But down where it's saltier, there might be other big predatory sharks that could maybe put a little bit more risk on them or could maybe snack them up. It might also be more food there. So I'm going to show you a video here, and it's going to be very, very chaotic, but I will talk you through kind of what's happening as it's going on. So we take all that data, we download it, we put it on a map like this, and this is what my sharks are doing. So you might notice the title of this is Resident versus Commuter Sharks. Those are the two kind of broad things we're seeing happen. So a resident shark is a shark that's going to be staying up there in those safer upriver areas. So you're seeing some sharks maybe just darting around those upriver areas. Those are what we would call resident sharks. And then some of the sharks are moving kind of up and down, right? So there's some that are moving really far down towards the lower, more downriver areas, and some that are moving really, really far up to those sort of upriver areas. Those are what we call commuter sharks. And as the sharks age and as the seasons change, we see different sharks do slightly different things. So you saw just then a lot more of them started kind of moving downriver as we move more into the wet season and leave the dry season. So that's one of the things that really informs our questions is we're seeing these two different sort of broad movements for these sharks. I want to know why. We want to know why a shark might be choosing or be designed to be a commuter shark versus a resident shark, and what they actually might mean for the system in general. So one of the useful things that we can examine from this is that these commuter sharks, the ones that are kind of traveling those really far distances, this is about 30 or 40 kilometers or so that they're traveling from the far upstream areas to the really, really salty downstream areas. And as they're moving, they might be eating those freshwater fish in the upstream areas, traveling all the way down to the marine areas, and then pooping them out. And then this reverse is also true, right? They might be down there in those marine areas, that ocean area, kind of eating those fish down there and swimming back to where it's fresh and pooping them out up there. And so they're actually linking these habitats that might be otherwise only flowing one direction. The sharks can go whichever direction they want. So it's really important and really useful for us to kind of look at what they might be eating and see how we can. I think of this as almost contextualizing that diary, right? Like we know what they were, where they went that day, but now we can also kind of figure out what they might have been eating on those days. So some samples we can take to check, right? We can take a fin flip, we can take a muscle sample, we can take a little bit of blood, or we can actually directly just take a swab of poop and see what DNA might be in that poop to see exactly what they might have been eating. And so this is what those samples kind of are broadly going to look like. This is how we actually go out and collect them. So we'll take that fin clip, we'll take that muscle sample, we'll take a little bit of blood, and then again, that fecal swab or that poop swab. We can bring those back to the lab and analyze them for things like DNA, for things like isotopes. And those are just sort of broad techniques that we can use to actually get down into what those animals might have actually been eating, and have sort of another tool to look at the ways in which they're moving relative to the ways in which they might have been eating, or the exact things in the case of a poop swab that we saw them eat. So that's another way of sort of contextualizing the diary for passive tracking. There's another type of tracking that I mentioned at the start that I'm going to talk about for the rest of this talk here, and that's active acoustic tracking. So all of the stuff that we just mentioned is sort of passive monitoring, right? We can see kind of from their chemistry what they might have been eating. We can see from these detections every, if they swim within 500 meters, where they might have been within that system, and we'll know when. But active acoustic tracking lets us actually follow that animal specifically. And this is a type of thing called biologging, which is basically instead of going inside of the shark, these go on the fin of the shark, as you can see in that far left photo there. And we can kind of build into these little biologger packages whatever it is that we want to measure. So in this case, for active acoustics, the thing that we're most interested is that sound producing device. And that's, if you look at the middle photo there, it's that, again, about the size of your finger, black tube that's kind of sticking out. That's what sends out the sound. So we can take that, we can stick that on the fin of a shark and follow it, listening for this sound. So this, wait for me to turn it on, is what the shark sounds like. And so if you hear that beeping, that's the sound every 1.5 seconds of that tag. And as I move it farther away, it's going to get quieter. Now we can't hear it anymore. So as we're following a shark around the Everglades, we're listening for that sound. We're monitoring sort of how loud it is and how we're hearing it. And we're adjusting our tracking based on that. So optimally, we can follow that shark for an entire day, maybe even 2 or 3 days. And that's exactly what we did. So last April, we took this exact tag package you're seeing right here and put it on a shark, Shark 531, that we have affectionately named Snickers. So we tagged Snickers, the bull shark. She was about a little under a meter and a half. This is maybe a 3, 4-year-old bull shark or so. So we tagged Snickers, we released her, and we tracked her for a little over 48 hours straight. And this is what tracking looks like. So there's that beeping again. There's Lucas recording all of the data as we're going. I was driving the boat. And we were also, beyond just sort of measuring where the shark was, monitoring a lot of water quality things. Things like temperature, things like salinity, and then most importantly, things like current. So how much water that shark is actually fighting with or against as it's moving around the Everglades, which lets us, again, contextualize these movements a little bit better, understand some of the reasons. And this is what Snickers did. So we wanted to kind of look at why this shark might have been moving. So again, we can zoom in on Southwest Florida here. That white dot, we're going to zoom in a little bit more here. So that white dot is where we tagged the shark. Again, it's in that upstream freshwater kind of safe area. So we got this shark tagged at around 7:23, released PM, I should say PM. It was very close to sunset. So got this shark tagged. She hung around in the bay during the day and every night started kind of moving down to these salty areas. So you can see on night one, that red area, that's where she turned around on the first night. And then that blue turnaround is where she turned around on night two. So 2 nights in a row, we saw this shark go from that safe freshwater upstream refuge all the way, almost all the way to where the Gulf starts, maybe like two-thirds, three-quarters of the way down, and then turn around in the morning and come back. So by analyzing that sort of data, we can figure out what sort of current she might have been swimming with or against. The tide was actually falling as she swam out, so that's an interesting thing there. We can also see kind of what the water qualities might have been like, where the shark was hanging out during the day, where the shark was hanging out at night, and figure out some of those reasons as to why this animal might have been moving when they did. There's some other cool things we can do with this active tracking as well. So within that biologger package, that it was pink in the photo with Snickers, now it's either orange in this photo here. We can put all different kinds of other things in there as well. So on the far left, that's a speed sensor. It's an accelerometer, just like in maybe your Apple Watch or a Fitbit or your phone. It also has like a little propeller on the front. That's that speed sensor. And it can measure all different kinds of things. But most importantly, it can measure when that shark is accelerating and when it's turning. So we can measure things like that. That smaller one that looks kind of like a little pencil eraser in my hand there, that one's actually got what's called a magnetometer in it. So it shows orientation relative to the North Pole or magnetic north. And that also lets us kind of figure out how this animal might have been moving in 3D space, as well as an accelerometer to again see how that animal was accelerating. And this is just another way of sort of contextualizing that diary entry even further. This is sort of like at the end of the day, taking the Apple Watch or the Fitbit off of the shark and downloading all the data to see, if it was really working hard that day or taking it kind of easy. Just a little bit more of ways to actually figure out what these movements mean in terms of what the animal is doing, and get into what those animals, why they're doing the things that they're doing, and not just where they're moving. And just like I mentioned on that far right photos there, that's measuring all that water quality stuff. So we can link movement, we can link energy, and we can link what's happening out there in the Everglades to really get a holistic picture, this kind of complete image of what these animals are doing. And then there's some future stuff with this that's really cool as well. I know that 3D printing is now like a really big thing. It wasn't sort of when I was an undergrad, and it's thankfully really big now. So what we did, I mean, massive credits to my friends Chase and Dr. Ben Bender who helped us down or create these sort of packages here. So they 3D modeled everything in space. We just put this on the 3D printer, and what used to cost, thousands of dollars to have these custom made by a company, we can do for like 25 cents. So we can design them custom for whatever we sort of want, whatever different things I want to be able to measure from that shark and kind of put on the fin. We can work with these designers and get everything really figured out. And then the last really cool thing we can do with this movement ecology is collaborate. So it's never one of just it's not just one scientist out there in the ocean collecting data on these one sort of sharks. We're part of this broad network called the Ocean Tracking Network that we contribute to. So anytime one of my receivers gets detections from any animal, not just my tagged animals, we'll download it and then upload it to this massive server so that any scientist, even if like a shark was tagged in, Jacksonville or West Palm Beach and it came out to the Everglades, I'll be able to detect it and let those folks know. And then vice versa. If my sharks come around to West Palm, I'll be able to get it from their receivers as well. So with new technology and these sort of big collaborative ways, we can answer even broader questions beyond just what's happening with my sharks. I think that's a really, really cool thing. That's sort of the future of all this movement ecology and really, really reading those diaries super well. So with that, I just want to thank all of the folks who've made this research possible with various fundings, various people who've helped me out in the field, all the people who've put up with me as sort of a person hopefully leading long, fun, hopefully, days in the Everglades. And I'm happy to take sort of any questions for the rest of this chat. Thanks so much.
Thanks, Will. That was really fascinating. It's great to learn about the research on bull sharks happening in the Everglades. We're going to transition now to Q&A, question and answer portion. Folks, remember you can put your questions in the chat box and I will work on getting those to Will so that we can get some answers for you. I'm going to go ahead and kick things off with a question where people want to know how did people, that's what they're asking, how did people
Mhm.
move the water, the historic flow of water to the way it flows now? That's a really good question.
Yeah, that's a really important question. And I think it's super neat because actually being from Jacksonville, this was never something I was aware of. Like I was in North Florida, I had no idea kind of what was going on in South Florida. And as we sort of settled South Florida, right, we needed water for the cities. So Lake Okeechobee is this massive freshwater body of water that we diverted to major cities to be able to supply, our drinking water, water for our showers, all that kind of stuff. And we realized kind of as we did that, that we weren't considering holistically the environment. We weren't thinking of how that might affect, the wading birds and the fish and the sharks and all the things that were already reliant on that fresh water. So we built all these sort of canals. You might have seen freshwater canals as you're driving past sort of any of these places in South Florida if you happen to live down here. The same way that we sort of diverted the water away, we can now divert the water back down. So there's timed releases. They know exactly how much water is leaving Lake Okeechobee. They can actually control when they want it to flow down. So it's timed best for wildlife. And there's a whole bunch of organizations, the Army Corps of Engineers, SERP, a lot of different government organizations that are really on top of this nowadays because we've realized how important this is. So it is kind of like a nice success story that this restoration project is actually having good things happening, and we're seeing really positive results from it. So it takes a lot of canals and a lot of folks working together, but we are actually starting to see positive things come out of this restoration.
That's great. And that kind of leads
Mhm.
into Ms. Tebow's class question. Regarding that, are there any negative effects that are still being noticed from that diversion?
Yeah, yeah. I mean, it's definitely not done yet. We've just finished, I think, the first major checkbox of this restoration, and it actually happened over in Naples. So it's called the Picayune Strand Restoration Project, and that was sort of the first big checkbox of finishing this restoration, which means that there's a lot of habitat that's not fully restored yet. We still kind of are hoping to get back to what that natural freshwater flow is, which is really important for my research because lots of those sort of upstream areas are still not getting the fresh water that they need to support the populations that we would like. So we know that there's more fresh water coming, and looking at sort of the ways in which that freshwater and saltwater matter for big top predators like sharks in my research is one of the reasons that it's very useful to be able to model what it's going to look like when we finish all these restorations.
Gotcha. All right, moving to a shark-based question here.
Mhm.
Miss Stanhope's class wants to know, do the tags stay on the sharks forever? For instance, when they die, do you get those tags back? Are they absorbed into their bodies? How does that work?
Ooh, that's a good question. So it depends on the types of tags. There's a couple different tags we can put in sharks. The two that I really focused on are those sound producing tags. So there's two different types I talked about. One is the internal one. So it depends on the battery life of the tag. Those will last for the entire animal's lifetime. They'll just stay inside the shark. Usually it's like 7 or 8 years. We're seeing even longer that they're actually pinging. So that's a good portion of the animal's life. That's close to half. So that's pretty good. That's pretty good for the sound producing ones. The other ones, the ones that we're putting on the fins of the shark, those are temporary. So those will just pop off and float to the surface and we can pick them right back up. The really
Interesting.
cool thing is how they release. So in salt water, they use a little piece of metal. It's called a galvanic timed release, which basically just dissolves in salt water. It's a fancy way of saying it dissolves in salt water. I don't work in salt water. I work in fresh water. So the ones that we use are this really like over-engineered piece of equipment, but it's super cool. It's got a tiny little bit of gunpowder inside, and underwater, it basically just breaks after 48 hours and pops harmlessly off the shark and floats right up to the surface. So that's how we
Interesting.
get those back, and we can know to the second when it's going to pop off on those.
Wow. Okay, so Elizabeth asks, speaking of the sharks that you're working with, how come the juvenile bull sharks are more tolerant to freshwater than the adults? That's a great question.
Mhm. Yeah, that is a good question. So there's two kind of notable things about that. One is that bull sharks are really the only species that are going to be fully tolerant of fresh, other than like a few other rare shark species. But here in Florida, they're the only one. So they can go all the way upstream and sort of hang out there, and it has to do with how they're transferring oxygen and salt across their gills, right? It's so that it has to do with their gill anatomy. And that makes them really, really safe from predators up there. Now, the other thing to note is when you swim in the ocean, right, you float a lot better than when you swim in like a bathtub or a pool, right? And the same is true for sharks. So they float a lot better when they're out in salt water, which means they're actually spending more energy hanging out in freshwater in the Everglades. And a big adult bull shark they're way more massive. So it takes a lot more energy of them swimming to stay afloat. but a little baby bull shark is smaller, so they're not spending as much energy to float. So when the big pregnant females come into freshwater to pup, they basically just go straight upstream and then they get straight out of Dodge. They're there as minimal time as possible because it's so energetically costly. But the babies, they can hang out there for a while until they get too big, and then they're not getting enough food, so they got to leave. And then, they're big enough and strong enough to go and compete sort of in the ocean.
Never
So
really
it's
thought
different.
about the energy expenditure. That's very interesting. Very
Yeah.
interesting. Great question, Elizabeth. Okay, we've got one from Julie too. She's asking why the sharks are more attracted to salt water at the nighttime versus the daytime.
Yeah,
That they were
there's
going
a
out toward, yeah.
so it could be there's a couple different things it could be. One is just like activity of prey. So there could be more active prey at that time that can get more food easily. Another one could be risk. So depends on predators, whether they're using sound cues or visual cues, but at night it's generally harder to be detected. So even if you're a little baby shark, you want to go down when it might be the safest. Which, if you're worried about getting eaten by a lemon shark or a tiger shark, nighttime's going to probably be a little bit safer for you. But it could also be related to other factors. One of the things I'm trying to look at with my dissertation is tides, whether they're kind of riding those tides down and back to save even more energy. So there's a lot of different factors that can influence when these animals are going to move. And historically, we've seen that it has to do with time of day. So they kind of leave at dawn and they come, or leave at dusk and they come back at dawn, which is exactly what we saw with Snickers. So that's kind of interesting. But there's other factors happening at play as well. So we can kind of build out these big models to figure out, what's most important for these sharks, especially because we've got 20 years of data to look at to do so.
Which is really great.
Yeah.
Kind of going off of that same question, Beatrice was wondering regarding the moving upriver and downriver, could it be also something to do with the water flows or temperature that maybe affects their pattern?
Yeah, there's so many different. That's one of the reasons that I wanted to look at holistically, sort of as many things as we kind of could, because there's so many factors that we know influence one or when where these animals might be. A big one we've seen in this system before is actually dissolved oxygen. So just like we need oxygen to breathe, and sometimes if you fly to Colorado, it's sometimes harder to breathe with less oxygen available in the air. It's the same deal for fish and animals that live in water. So if there's less oxygen dissolved in that water, it might be harder for them to be able to breathe. So dissolved oxygen is a big one. Temperature we know is a big one. I've been a little bit worried about my baby bull sharks this past week in South Florida. It's been a little chilly,
Yeah.
so I've been had the water station open, monitoring and looking. And like, okay, it only dropped 1 degree Celsius. They're probably okay. So temperature is a big one as well. I know it's on all of our minds this week. So yeah, that influences where they'll hang out. If the temperature gets low enough, sometimes they just leave. Sometimes they'll just leave and go find warmer waters.
Oof, I bet they were a
Mhm.
little chilly. So
Yeah.
Miss Page's class was wondering if you've ever dived with sharks during your research. Do bull sharks lay eggs or do they have live births? And how do you monitor this? That's a great question too. A couple
Yeah.
questions there.
So we'll start with the first one. I've definitely dove with sharks before. I've never dove with my little baby bull sharks because I like to describe the Everglades as ranging from iced tea to chocolate milk in terms of visibility. So there's no real thing to gather from diving with them down there. But in the various places I've worked, I mean, we've dove with sharks. You see nurse sharks and everything in the Keys all the time. So that's super neat. The next question I want to take from that one was whether they give live birth or whether they lay eggs. So they are ovoviviparous, which means that's a big fancy term for saying that they basically keep the embryos inside of them and then they give birth to a fully formed shark. So just like we have like a belly button, and that's where the umbilical cord is attached to us. Baby sharks, and this is the way we actually can tell if they're in the first year of their life, they'll also have an umbilical scar. So it sits kind of a little bit higher up on them. It'd be kind of like up here on us. And if it's open, it means that they were born, within a couple of weeks, right? So we'd call those neonates. If it's closed, but it's still visible, they were born that year. So we'll call those young of the year. And if you can no longer see that umbilical scar, it means they're about a year plus in age. So they're a little older than a year. So yeah, they do kind of give birth to these live individuals. And that actually helps us figure out how old some of the sharks we're catching are.
Fascinating.
Mhm.
So PK wants to know how are you catching the sharks to, for example, look at age? What are your methods for doing so?
Yeah, so there's a couple of different methods we use in general for shark science. The one that I'm using in the Everglades is what's called long lining. So if you've ever had like a lot of commercially caught fish are also caught on long lines, and it is exactly what it sounds like. It is one very, very long line that we just clip a whole bunch of hooks onto. Commercially, these can be like 12 to 15 miles long, even, but mine are only 500 meters. So we put 50 hooks out. We put them about every 10 meters or so, and we just stick on the end of a nice circle hook, a big piece of mullet. And that helps us catch. We get like one shark every line, maybe like a little less than one shark every line on average. So we do pretty decent. It gives us a nice way of consistently monitoring these populations. And we've been doing this for almost 20 years too. So we've got this. One of my friends in our lab is looking at that long-term catch rate or rates and seeing kind of again what factors might influence when we're catching a shark. So lots of cool things we can do with data beyond just tagging these animals as well.
Interesting. So there's a few classes or students from a few different classes that are asking similar questions regarding the tags once you've caught them. They seem like they're pretty large, and they were wondering if it impacts the shark at all with the way they swim, or does it bother them? And approximately how long those stay on for, too.
Yeah, yeah. So it's interesting. You wouldn't necessarily think about it, but like us. So I don't know if you can't see all of me, but I'm pretty solid, right? We're solid all the way through. We don't really have any empty space in our bodies, right? We kind of fill that out. Sharks have an empty body cavity. And so when we're actually taking these tags and doing that like super quick, like 5 minutes in and out kind of surgery, it actually just sits in this empty space in their body cavity. And it just kind of hangs out there, right? So it's not actually blocking anything. It's not impacting anything inside of them. It's not like pressing up against any of their organs. So we put it kind of in that empty space, and it just sort of sits right in there. And also these are animals that are underwater, so there's a lot of hydrostatic pressure, we call it, that's also going to be kind of on these animals. So they, from what we're seeing in the literature, they heal incredibly quickly, and it doesn't affect their sort of behaviors at all. And they heal. We do other kinds of tagging too. That's useful. You might have seen I read off Snickers's tag number. So we put tags on their dorsal fins as well, so we can ID them if we ever happen to recatch them. And they don't really have any nerve endings in that dorsal fin either. So it's sort of even less than getting your ear pierced. They sort of barely feel that, which we want to be. It's really important, like in science, that we understand how we might be impacting these animals. And so we have to report what we're doing to sort of a committee that keeps track of everyone at the university. And we all have to justify and make sure we're doing things in the most safest, efficient, and most humane possible way for all these animals. So we keep that in mind as we're designing these questions, and we always want to make sure we're following up and keeping track of all that.
Very
Mhm.
good. Okay, so there's a question about the poop swab. They want to
Yeah,
know, are you seeing diet changes at different times of year or where they're hanging out in the water from the poop
yeah,
swab?
that's a great question. That is actually the thing I am currently working on because I am giving another presentation this weekend on the poop swabs at our university here. So yeah, we are seeing sort of these differences in what they're eating based on age class and based on season. So wherever they're hanging out, they tend to be eating things directly there. I'll tell you the things they eat the most are in line with some of the previous research we've done sort of in this system and other estuarine systems in South Florida. They're eating a whole bunch of mullet and they're eating a whole bunch of catfish. And those are sort of the fish that are found broadly all across the system. So that makes sense. Like wherever they are, they've kind of got access to some of those fish. But one of the really neat things is in the Everglades, we have this freshwater marsh that's putting in a lot of freshwater prey that isn't going to be really accessible down in the marine areas. So, like the Mayan cichlid is what I showed earlier in this presentation. And we're seeing a whole bunch of those kind of fish showing up in our shark's diet in the dry season, which is really important because that means that as those marshes are getting dried down, there's less rainfall and less water flow coming from Lake Okeechobee. Those fish are getting forced into the Everglades, which is this nice food source for bull sharks. So it really depends, right? It depends on the time of season. It depends on like a lot of other environmental factors, but we are seeing that they're preying on these slightly different marine versus freshwater fish depending on that season, which is really cool. That does mean that they might be linking these habitats.
Very interesting. All right, so there's a question from Colin and Candace. They're asking something similar, so I'm going to kind of group these together. They want to know what impact the signals from the trackers emit, or what impacts do those tracker signals have on sensory systems of sharks? Meaning, are they picking up electrical signals or
Mhm.
anything from those tags, I suppose, is what they're asking.
Yeah, that's an interesting question. That's something that there's actually some pretty neat research on. So they are all at off frequency tags. So or they're all at different frequencies of tags. So we kind of know what frequency they're emitting at. And research can be done to kind of test on whether that frequency might be, even auditory or even able to be heard by these animals. And one of the things I think that really inspired this type of research is they were tagging seals, and they were kind of worried that if you're tagging a seal, A great white shark might be able to hear that seal as it's swimming around and basically just have like a beacon for where its food might be, sort of like a big dinner bell ringing to come, eat me, right? And what the research is showing is that it's not transmitting at a frequency that's detectable by these marine life, because if it was, the shark would immediately come and swarm that seal because they could just, again, hear the dinner bell kind of ringing. So it's not transmitting at a frequency that should be auditory for anything. And for us, you can feel the vibrations. It actually works by these little ceramic plates kind of banging into each other. And if you press it against your temple, you can feel it kind of vibrate a little bit. But like we can't hear it either. It has to be. That's why those hydrophones kind of listen for it. So it's not impacting them in any negative way that we really know of. But it's able to transmit at a frequency different than what should even be auditory, at least
Very
as far as we
interesting.
know currently.
Yeah, yeah, that's really interesting and a great question, Colin and Candace.
Mhm.
Um, the audience in general wants to know what's the average life expectancy of a bull shark and do they have any natural predators?
Yeah, yeah. So it's going to be 20 years, give or take, is kind of the lifespan of bull sharks, and then predators are going to depend on where they're at. So there's been some wonder if maybe gators are eating these bull sharks in the the Everglades. We don't have a whole lot of evidence to suggest that that's happening regularly, but it could. More so, it's going to be bigger sharks eat the smaller sharks. So as you're a baby little juvenile bull shark, if you happen to go down into those more marine areas of the system that we talked about, there's big lemon sharks that could be there. There's, other big adult bull sharks that could eat you. There's even tiger sharks as you kind of get out into the ocean. And generally, if the sharks can fit it in their mouth, that's what they're going to eat. So your main risk is anything bigger than you that is going to potentially just eat you.
There's a question about the number of sharks that get tagged collectively, and what is it considered like, I guess, a good number for data collection to make the data meaningful?
It really depends on which species you're looking at. So for us, we have, I believe, the longest running sort of tagging program like of this kind. So we've got 20 years of data, which is really, really cool, and 197 total tagged individuals across all these different timescales. So because these animals are staying in the system for 4 years or so, we're getting really good data of what's happening across all these different years. If we were studying something like, oceanic white tips or an animal that's going to be traveling really, really far ranges and be pinged intermediately, we might need more tags than what we currently have right now. But for us and for our system, because these animals are staying here so long and we're getting such consistent data for, multiple years of their life, this is a really, really robust, like really awesome project that I'm really glad to have been able to sort of tack on to the end of here and contribute to, again, 20 years of research at this point. So we've got pretty good sample sizes for what this is, but it all depends on the species and the questions and the things you're trying to look at with that.
So not only do we have elementary kids, middle school kids on the call, etc.,
Mhm.
we have a couple big kids on the call too. Grain is asking, they're interested in getting into this field and they
Mhm.
wanted to know how you analyze the data. Are you using R for doing so? Are you
Yes.
all, for the folks that don't know, you might want to explain what R
Mhm.
is.
Yeah, yeah. So there's different ways you can analyze data, right? You can kind of get it organized and figure it all out in Excel. But like I just mentioned, we're working with really long-term data. So my detections from all my receivers are over 50 million. So if I have 50 million rows in Excel, there's no way, or anything, right? I can't read 50 million things. It would take me 20 years to just read all 50 million. So R is this really kind of awesome program that's open source. Anyone can design sort of anything they want to be able to do in R, and you can kind of write code. So it's this program where you write your code, and that code will do whatever it is you need to do. So in my case, my data is way too massive to analyze in any other system. So me and R have gotten real friendly, and it's one of those skills that I think if you want to get involved in higher education and you want to go to graduate school, even undergraduates at this point now, it's a really useful tool. That as much as we probably don't love AI, and I think a lot of us see the problems with AI, as artificial intelligence is improving, it really is another tool that we can use to make coding accessible to everyone. So like I'm not like a computer person, like I'm not, as much as I like video games and movies and things, I'm not going to ever be able to code one. But with R, even a skill like that that's kind of above what I understand, it becomes, or with AI, R becomes accessible. So highly recommend, like if you're in college or you're thinking about graduate school, start thinking about R, start thinking about some of those skills you might want to learn, and talk to folks like me who can maybe point you in the right direction and give you some advice on where to use them and how to actually use them effectively and correctly. But yeah, R is
Great
great. I love
tips.
R.
Yeah, great tips. Okay, we have a question from Miss Guimare's class. They're really interested in Snickers and wanting to know if Snickers is still being tracked and how they can follow, if there's a possibility to follow. I
Oh,
think that's great.
Snickers. Oh, Snickers. Yeah, so like I mentioned, these tags don't stay on the sharks permanently, so they pop off after 48 hours or so. We had a little bit of difficulty kind of recovering Snickers' tag after it popped off, but there's no way to actively track Snickers sort of anymore. These are sort of confined, really quick, one weekend kind of active tracks. However, all of my other passively tagged bull sharks are still out there collecting data. We've just got to go out there and collect them. And the really cool thing about these active tracking and these biologging things is the data is so dense. There's so much data. Like 50 million like lines and detections is nothing in biologging because it measures things like 20 or 50 times a second for 4 days, 3 days straight. So the data is so dense that there's no way to offload it. It's not like a satellite tag where we could beam it up to a satellite, or these hydrophones that we can kind of connect via Bluetooth. We've got to physically pick that data back up and then just plug it into a computer to download it. So we can't follow Snickers remotely anymore. She's still out there in the Everglades doing whatever Snickers is doing, hopefully swimming back and forth on that Harney River. But the data
Fantastic.
is just so dense.
So one of our groups must be in closer proximity to Merritt Island. Ms. Ellie's group is wondering if bull sharks are up that way near Merritt Island too.
Yeah, yeah. I mean, they're pretty ubiquitous. Bull sharks are found sort of all over the world, even. And any coastline, any nearshore areas, any estuaries, you might have little baby bull sharks in there. They're going to be hanging out there. Just anywhere that's going to have some freshwater, saltwater mixing. There might be some baby bull sharks being born there. There's actually a project that a friend of mine in another lab across the hall is working with pregnant bull sharks in West Palm Beach, actually kind of tagging them to figure out exactly where and when they're giving birth. So anywhere you've got this freshwater, saltwater estuary mixing, there's probably going to be some little baby bull sharks hanging out there, which is great. These are really important predators that contribute, globally to these marine food webs. So we want nice and happy, healthy bull shark populations. It's a good thing.
Fantastic. Ms. Stanhope's group wanted to know, and you might have stated this, but I have the question again about when they reach maturity. I know you talked about the different stages and how you can age them, but at what age are they considered mature?
It depends. So it's different for males and females, and it's more about sort of size, right? So when a shark reaches maturity, we have these different tables for different species that basically say, if it's a female shark, it's really hard to tell, right? You have the size and you have an age, and you say, okay, if it's above this size and age, it's probably, a fully mature female shark. Male sharks, you can actually ID from females based on these sort of external fin appendages called claspers. And if those claspers are calcified, it's just sort of a visual way of looking at the animal and saying, yeah, that's a mature male shark. So in bull sharks, probably like 12 years ish, give or take. Again, it depends on males and females, and it depends on sort of how much food they might have been getting as juveniles. But we can actually figure out, where they are based on, size and age. Yeah.
Very good. Okay, so I think we're going to wrap it up with one question that's got a
Mhm.
couple parts to it. How would you, if you're interested in sharks and shark research, how would you recommend folks get involved? What advice would you give them if they're interested in a career like yours? Are
Mhm.
there particular classes or other experiences that you would recommend to do so?
Yeah, I love this question because I think it's one of those things where science really needs to be approachable, right? And that's the biggest advice that I can give anybody that's even remotely interested in this is just talk to folks. As you can probably tell from the way I'm answering these questions and hopefully from my talk, we love talking about our stuff. It's like our favorite thing to do. So talk to scientists, talk to graduate students, talk to folks like the Anjari Foundation and local nonprofits that are involved in these things. Go to the parks, anything sort of local in your community. I promise there's something marine science, environmental science, any of those types of things. So getting out there, getting involved and talking to folks lets you figure out kind of what's going on, and then you can get involved in those things. So as an undergraduate, I ended up with these awesome scholarships that let me go to South Africa and work with these great white sharks and fall in love with this field just because I was talking to folks who told me like, hey, this is available, you should apply for it. So the more you can kind of get involved, the more you can talk to people, the more you can really get your feelers out there to know what's going on in the field is going to help you inform better what might be a good fit for you. What classes to take, right? When you're an undergraduate, if you're ever, pursuing higher education or even in high school or whatever grade level you might be at, volunteer with folks, talk to graduate students at your universities. I promise there's a tired, sleep deprived graduate student somewhere at whatever university that needs help with whatever project they're doing. Talk to them. I have a small army of interns that I take out into the Everglades to tag bull sharks. And why couldn't that be you, right? So if you go to a college, find anybody who's involved, talk to them, figure out what kind of research is going on, because I promise we not only love to talk about our stuff, but we love to get people involved in our stuff as well. So that's the biggest advice I can give you is just send that awkward email, reach out to people, and I promise like it's going to help you in the long run.
That's super great advice. Thank you so much, Will, for sharing with us about your project. I'm looking to see what's going on with the slides. There we go. I wanted to quickly thank you, Will, and thank everyone for tuning in today and taking the time to learn about sharks and movement ecology in the Florida Everglades, these wonderful bull sharks. There is a recording of today's talk that is available at the UF Earth Systems YouTube channel. For more information on the Scientist in Every Florida School program and Jerry Foundation, you can follow us, visit our websites, follow us on social media. Today was the first of our 2026 spring series. I wanted to share with you our next live stream webinar will be on March 4th. That'll be at 9 o'clock as well. We'll be speaking with Matthew Anderson of Indian River Lagoon Aquatic Preserves. He'll be sharing about the work he does with American oyster catchers in the region, including bird banding and how they collect the life history data of these birds, their movements, mate selection, populations, and more. And then on April 1st, we will be wrapping up the spring with a lineup from Alexa Elliott. She is with the South Florida Public Broadcasting Service. She'll share her Emmy award-winning Changing Scenes Marine Science documentary series and how they produce that from start to finish. So in the meantime, on behalf of Scientists in Every Florida School and the Anjari Foundation, we again want to thank you for joining us today, and we look forward to seeing you on another Ocean Expert Exchange. Thanks, everyone. Take care.